Hello and welcome to the Nobel Forum at the Karolinska Institute in Stockholm, Sweden. My name is Göran Hansen and I'm a member of the Nobel Assembly that awards the Nobel Prize in Physiology or Medicine. We're standing now in the Wallenberg Hall of the Nobel Forum, which is the hall where the Nobel Prize is announced every year in October. As you know, this year's Nobel Prize in Physiology or Medicine was awarded to Robert Furchgott, Louis Ignarro and Farid Murad for their discoveries concerning nitric oxide as a signaling molecule in the cardiovascular system. This research was carried out from the mid-70s and more than a decade further on, and resulted in the identification of nitric oxide as a signaling molecule, which was a big surprise because before this research, nitric oxide was known only as a pollutant, really. We're now going to take a look at a video that shows the function of nitric oxide in the cardiovascular system. The first slide that you see here is simply a cartoon of the heart. The heart with its coronary arteries are shown in the following video sequence, and you can see that there is a pulsation in the coronary artery. This pulsation, of course, is caused by the contraction of the heart, which propagates the blood into the coronary artery. And the coronary artery can dilate. It can widen itself when the blood flow comes. That means that it's elastic. It also has a capacity to regulate its own diameter. It can control its own diameter by active signaling mechanisms in the artery itself, in the arterial wall. And that's the function of nitric oxide, together with some other molecules that have been identified. Here we see a cross-section of the coronary artery, and you can see that there is a thin layer of cells that face the blood and form a border between the blood and the vessel wall. That's the endothelium. Under the endothelium, you have a thick wall of muscle cells organized circumferentially. These muscle cells can contract and relax. They are a fairly primitive type of smooth muscle cells. The arteries, as you know, is the site of one of the major lethal diseases in the world, atherosclerosis. Here you see an atherosclerotic plaque that has developed in the coronary artery. This, of course, reduces blood flow through the artery and can cause significant problems with ischemia, especially when the demand is greater than the supply, in exercise, that is. It becomes even worse if there is a contraction of the artery. That can stop the blood flow totally. The patient develops symptoms such as angina pectoris, and if the ischemia becomes persistent, myocardial infarction. We've known for more than 100 years that nitroglycerin can stop this process and revert the situation. It does so by dilating the artery, by relaxing the smooth muscle cells. And as you can see here, when nitroglycerin is taken by the patient, it rapidly goes to the blood vessels, relaxes the smooth muscle cells, and dilates the artery. Blood can start to flow through again. The heart muscle gets the oxygen and nutrients it needs, and the symptoms disappear. It's an effective treatment, and it's been used for over 100 years, but the mechanism was unclear for many, many years. But thanks to the research carried out by this year's Nobel laureates, we now know how the system operates. And in fact, nitroglycerin mimics the action of nitric oxide, which is the endogenous nitroglycerin of the organism. Now we're going to go into the cellular details of the activity of nitric oxide. And in this picture, you can see the endothelial cells and the smooth muscle cells under them. On the surface of the endothelial cell, you see a receptor. And now an agonist will hit the receptor. This agonist could be a signaling molecule such as acetylcholine, for instance. When the receptor is activated, the endothelial cell starts to produce nitric oxide. Nitric oxide, as a gas, diffuses in all directions, But quite a lot of it goes down into the underlying arterial wall, and there it reaches the smooth muscle cell. Since it is a gas with hydrophobic solubility properties, it can penetrate through the cell membranes directly into the smooth muscle cell, into its cytosol, and there the nitric oxide can hit an enzyme, guanylyl cyclase. Guanylyl cyclase has a heme group that can be targeted by nitric oxide. And when that happens, the enzyme undergoes a conformational change. That change leads to an activation of the enzyme. And now guanylyl cyclase starts to use GTP to produce cyclic GMP. Cyclic GMP, one of the second messengers of cells, will in turn start a cascade of chemical reactions involving phosphorylation of different substrates in the cell. And the end result of that is a relaxation in the myosin-actin contractile apparatus of the smooth muscle cell. So when finally myosin is hit by the chemical reaction initiated by nitric oxide, the smooth muscle cell relaxes. Its contractile filaments slide apart and the cell relaxes and the whole artery dilates. Now, if this happens in all smooth muscle cells, of course the artery will dilate, the lumen will become bigger, and more blood can flow through the artery. It's obvious that if this happens in one particular region of the body, such as the heart, the result will be increased blood flow to the heart muscle. If it happens in the whole organism at the same time, the result can be a reduction in blood pressure. And in fact, nitric oxide is an important regulator of blood pressure. Now let's take a look at the contributions made by this year's Nobel laureates. Ferid Murad studied the effects of nitroglycerin and similar pharmaceutical compounds in the 1970s and found that nitroglycerin acts on guanylyl cyclase to induce production of cyclic GMP in the smooth muscle cell. He wondered how nitroglycerin, this big molecule, can act on an intracellular enzyme in the smooth muscle cell. And then he discovered that nitroglycerin acts by releasing nitric oxide, which is the active compound that goes into the smooth muscle cell. This was the first piece of evidence for a biological function of nitric oxide in regulating the cardiovascular system, in regulatory function in the human body. Murad proposed already in the 70s that endogenous signaling regulating molecules such as hormones may perhaps also act by releasing nitric oxide. But there was no more information about that subject for a few more years. In 1980, Robert Furchgott studied the function of the endothelium and the interplay between endothelial cells and smooth muscle cells. He then discovered that if you stimulate an artery with acetylcholine and the endothelium is intact, the artery relaxes. But if the endothelium is damaged, there is no such relaxing action. Instead, the artery may contract. The reason for that, he proposed, is that acetylcholine induces the production of a factor in the endothelium. This factor goes to the muscle cells and relaxes them. He called this factor the endothelium-derived relaxing factor, but its chemical characteristics were obscure for another 6 years. During those years, from 1980 to 1986, several pieces of evidence were coming together, but the story wasn't really complete until in the summer of 1986. Then at a meeting in Rochester in Minnesota in the United States, Louis Ignarro and Robert Furchgott both presented a series of evidence pointing towards nitric oxide. And they both proposed that nitric oxide is the endogenous nitrate that is the endothelium-derived relaxing factor. They had a series of evidence that they have accumulated in their respective laboratories independently of each other. With that, the first phase in the nitric oxide era was finished. Newell had found that nitric oxide was released from pharmaceutical agents, and he had deciphered its mechanism of action in the blood vessels. Then Furchgott and Ignarro could identify the endogenous nitric oxide, that is the EDRF, and its production and action on the smooth muscle cell. Several laboratories now continued and expanded the research. And it was found that nitric oxide is produced by an enzyme, nitric oxide synthase, which can be made in several different cell types, in the blood vessel endothelium, but also in neurons as well as in immune cells. And today we know that nitric oxide production by this mechanism is important in signaling not only in the cardiovascular system but also in the nervous system and in the immune system in inflammation. So the research on nitroglycerin and on signaling between endothelial cells and smooth muscle cells has resulted in the identification of an entirely new type of signaling molecule in the human organism. That is an inorganic small molecule, a gas that can transmit signals from one cell through another, through cell membranes, directly from the producer into the cytosol of the responding cell. And it's been truly a fascinating story that has emerged from the first findings in the mid-70s until the wealth of knowledge that we have today. Thank you.